Alrighty. Hello, my name is Anthony Martinez, and today I'm giving a presentation about histoplasma capsulatum, its biology, and its human interactions. You can see on the left here, this is a picture of the hyphal form of the organism that we're going to be talking about today. I think it looks rather beautiful. Okay, so to start off, we're going to look at its taxonomic classification. It is a eukaryote, obviously, since we're in eukaryotic microbiology, but it is a fungi. It is located in the phylum Ascomycota, and I just wanted to throw in a general life cycle of that phylum here. So if we look at this general life cycle for ascomycetes, we see a few interesting things right off the bat. The first is if you look at these teal arrows, it spends the majority of its life in a haploid form rather than a diploid form, which is what we're used to for like humans and mammals and that sort of thing. It spends a very small period of its time in an actual diploid / dikaryotic state. And then the second part that we can see from this is it has a sexual reproductive phase and an asexual reproductive phase. In the course of my survey of the literature, there wasn't much about a environmental sexual reproductive phase. These structures like the ascocarp and karyogamy and plasmagamy and all of that stuff isn't really well characterized for this organism. Instead, what we're going to be focusing on more is the actual asexual reproduction side of things. So a quick rundown of that. You can see that this is hyphal, not hyphae, multiple hyphae making mycelia. So if you look here, you see red and blue nuclei. So we'll follow this red one for now. There's red one. You have a spore. It lands on something. It germinates. It forms hyphae and then mycelium. Eventually, once it has enough nutrients, it forms a structure called a kinidiophore. The top of these structures is where the actual asexual spores are generated and released. For our purposes, we're going to be focusing more on microconidia. So for this, we'll just act like this is a microconidia. Once it's generated on this kinidiophore, it is dispersed. In our case, it sounds like it's dispersed in the air, which can then be inhaled. There are multiple mating types. So you can see this is a plus, and this is a minus. They meet each other, and then that's when you get the sexual reproductive phase of things. So this is a general life cycle of what one would expect to find in the phylum Ascomycota. Today, of course, we are working with histoplasma from the genus Histoplasma and the species Capsulatum. Alrighty, so the second part of this overview is the global and genetic distribution of histoplasma. So if you look on the right here, we can see an unrooted phylogenetic tree. Each one of these values is a bootstrap value. A bootstrap value of 1 or 100% is ideal. So you can see this is a pretty decent unrooted phylogenetic tree. Second thing that you can see is that there are quite a few branches here, which indicates there's quite a lot of genetic diversity. And the third thing that you can see is that it's widely distributed. So you have Latin America group A, Latin America group B, you have North American groups, Australia, Netherlands, Africa, Eurasia. So it really is distributed worldwide, and it has quite a bit of genetic diversity. Even with populations that are in the same continent, you can see this is Latin American group A and this is Latin American group B. They're quite distant from each other, genetically speaking. And if you look over on the left, you can see the worldwide geographic distribution of this organism. It's mainly found in North America, specifically the Ohio River Valley and Mississippi River Valley, though that's based on data from like the 1950s. So whether or not that can be trusted today is kind of, well, not known because the data doesn't exist. It's also found in South America. You can see pockets in Europe, West Africa, a little bit of Central Africa and South Africa, and then in Asia, of course, in India, China, and looks like Indonesia here. So variants of H. capsulatum are found kind of everywhere. In terms of the actual local environment where you would physically find this organism, so say if I was in South America and I wanted to find this organism, where would I be likely to find it? It would be found in soil that has been contaminated with bat or bird feces. It's found in many mammals, including dogs, cats, badgers, hedgehogs, that sort of thing. But it didn't seem like their fecal matter was highly correlated with human infection. So it seems like the sources for that, at least, are mainly avian and bat in origin. The bird and bat feces itself is actually rich in micronutrients, specifically nitrogen, which helps support the growth of this organism. And here you can see a typical route of transmission, and we'll talk a little bit more about that later. But in this case, a chicken has deposited fecal matter here that was contaminated with probably the yeast form of this organism. That is then inhaled, and that is where you get the establishment of histoplasmosis in the lung. Yeah, so it's found in a variety of different organisms, and those organisms can be found throughout the world. So that is pretty interesting. In terms of the microscopic morphology itself, this is an interesting organism because it has a dimorphic physiology. That just means that it has two forms. The first is a actual hyphal filamentous form, which we're usually familiar with when we talk about fungi. And you can see that here. These little spiked balls themselves, the microconidia, and then these filaments are the hyphae. Once these conidia become airborne, as you can see here, it's usually inhaled by, in this case, a human or other mammalian host or avian host. It makes its way to the lungs, and then the change in temperature from ambient temperature to inside of the mammalian, or in this case, human host, is actually the trigger event for the change in morphology. So it goes from this filamentous form to this yeast form here. And the yeast form is what's actually pathogenic in humans. In figure 6 down here, you can see this quite clearly. This is the hyphal/mycelial form of the fungi with these conidiophores located here. And then once it hits 37 degrees Celsius, which is the average human body temperature, it triggers a morphological or morphogenesis event, which changes the morphology from this hyphal form to a yeast form. Again, there are other genes and secondary messenger molecules involved in the transduction of the signal, but the actual signal itself for this morphogenesis is temperature. Now, once it gets inside of your lungs and it changes into its yeast form, it invades alveolar macrophages. So you can see that here. These are actually macrophages, and then which have been stained with. They haven't been stained. These are fluorescently labeled yeast form of the pathogen. You can see here that the macrophages are kind of throwing out these pseudopods, and these are quite clearly labeled in here. Once the pathogen is actually inside of the macrophage, will be found in the phagosome in the actual macrophage. So this presents with a few problems. The first is that in normal macrophages, iron and zinc are effluxed from the phagosomal environment, creating a very low concentration of both of those molecules, or I guess ions. And in general, the phagosome is a very nutrient-poor environment. So the first thing that it needs to do is it needs to prevent it from being degraded. The pathogen itself needs to prevent phagosome pH drops. And then it needs to also acquire important molecules. So one of the important molecules is iron. In order to get that into this environment, it needs to maintain a pretty close range of pH between 6 and 6.5. This is important because a pH lower than that will activate hydrolases, which will start degrading things. And it needs to be in this pH range for this molecule transferrin to be activated. Transferrin is located in the membrane, and then it basically just traffics iron from the extraphagosomal environment to the intraphagosomal environment. And then these cyridophores, you can see with these green stars, will actually latch on to that iron and allow it to be moved into the organism itself. De novo synthesis of molecules like riboflavin and uracil is incredibly important, but one of the interesting attributes of this organism is that it's auxotrophic in its yeast form stage for the synthesis of cysteine. And so what actually happens is they think that this has a high intraphagosomal concentration of sulfur or bioavailable sulfur-containing molecules, and that allows it to generate its cysteine without having to generate it completely biosynthetically. So yeah, that's pretty cool. Okay, so in terms of transmission of this actual organism, Thankfully, there's no human-to-human transmission, as humans are a dead-end host. As a kind of a side note, that's kind of a bizarre thing about this organism, is that it seems to be adapted for survival in macrophages, which are usually found in mammalian systems. But in mammalian systems, it seems to operate like a dead-end host. We don't really see um, reproduction or that sort of thing, at least not when in my literature review. So most infections that are human, most human infections are primarily acquired. So let's go over like kind of how that happens. So you have your reservoirs in the environment. In this case, they're saying uh, possums and bats. It grows in them in the mycelial form, but in temperatures between 18 and 28 degrees Celsius, typical humidity, that sort of thing. It's found in woods, caves, hen houses, uninhabited buildings, tops of trees, basically anywhere where bat or avian excrement can be found. Um, the actual microconidiophores become aerosolized, which is inhaled by the host. This traffics to the lungs, where it transitions to its yeast form, and then the intracellular yeast form is associated itself with tissue damage, and then that probably kills the host, and then that's what releases it back into the environment. There are two forms of conidia for this organism. There's micro and macro conidia, but for our purposes, micro conidia are the ones that are important for establishing pulmonary infection. So before I get into all these words, I kind of want to take a minute and look at this figure here. So this figure shows interactions between the yeast form of the pathogen and different innate immune cells. So you have your dendritic cells, your neutrophils, and your macrophages down here. The cool thing about this is that it changes its cell wall a little bit. So you can see in chemotype 1, you have this beta-1,3 glucan, right? And so this is what generally triggers TLRs and will initiate anti-inflammatory responses and that kind of thing. You can see TLR2 binding here and dectin-1 binding. CR3 is what's actually mediated. It was the receptor that this pathogen will actually bind to be phagocytosed. And then you can see in chemotype 2, this outer layer of red, which is alpha-1,3 glucan, right over the beta glucan. And that actually prevents TLR recognition of the beta glucan, which I think is pretty interesting. And then you can also see here VLA5 interacting with CYPA on the actual organism itself. And that is what mediates communication interactions between the dendritic cell and H. capsulatum here. So like I've kind of already indicated before, the microconidia are inhaled and they make their way to your pulmonary alveoli. That's where it changes into this yeast form. The higher body temperature of 37 degrees is what actually triggers this morphogenesis. Once actually in the macrophages itself, it can disseminate widely throughout the body because macrophages, of course, are members of the innate immune system, so they traffic to the lymph nodes. Because this is an intracellular pathogen in macrophages, they are also trafficked to the lymph node, and once there can be disseminated into the bloodstream, where they make their way to bone marrow, the spleen, the liver, etc. And like I said earlier, humans are dead-end hosts, but for some reason it seems that they've adapted many mechanisms to be able to invade macrophages and other innate immune cells in humans. In terms of the trigger events for morphogenesis, I kind of want to look into that a little bit more and kind of look at it in this light of its role in virulence and pathogenesis, because without this, you really don't get any pathogenesis from this organism. So this is really key in establishing infection and becoming pathogenic in human hosts. So you can see here in this great table, the hyphal form here. This is room temperature conditions, glycnac, some cyclic AMP levels and self-hydryl oxidization lead to a hyphal form. The actual genes themselves involved in this are right here. The genes that are interesting for actual morphogenesis are DRK1 and these RYP genes. So DRK1 stands for dimorphism regulating kinase, and is a homolog of this gene found in H. capsulatum. It's blocking the expression of this gene in H. capsulatum actually disabled the transition from from the hyphal form to yeast, and that also disabled the expression of associated virulence genes. And then, of course, the actual promoting conditions themselves are 37 degrees Celsius, in this case, a sulfhydryl reducer, and the presence of cysteine. Cyclic AMP levels in like the 70s in a paper were also associated with this transducing of the signal. But without these genes, these DRK1 and the RYP genes, you not only don't get a transition from the hyphal form to the yeast form, but you also don't get expression of the virulent genes associated with the yeast form. So these are really, really important and key. Unfortunately, this is kind of the only information we have about these guys right now. This organism really isn't well studied, and the actual specific molecular mechanisms of how these genes work, what proteins they make, etc. Are they making transcription factors? How are these signals transduced? That sort of thing have not been well elucidated. They really are only in the beginning stages of figuring this all out, which is kind of unfortunate because I kind of wanted to know. So another virulence factor is its ability to survive in the macrophage itself. So when the macrophage encounters the organism, it's going to start producing these reactive oxygen species, especially if that dectin-1 molecule recognizes the beta-glucan in the cell wall of the yeast. It's going to start producing these reactive oxygen species. And when it's internalized in the phagosome, it's going to further encounter these reactive oxygen species. So how is it going to be able to subvert this? Of course, it has genes and proteins involved in this subterfuge. One of them is SOD3. This is a superoxide dismutase, and it's shown to be upregulated 100-fold in the phagosome. And you can see here that kind of takes out this reactive oxygen species here. You can whatever. And then this is hydrogen peroxide. You can see that CatB is involved in destroying that. The cool thing is not only does it have these two proteins in the phagosome, but if the hydrogen peroxide makes it inside the yeast cell itself, it has a secondary protein here called CatP, which will destroy that hydrogen peroxide if it's accidentally taken up inside the yeast cell itself. So these are some of the mechanisms it uses to prevent destruction by reactive oxygen species once it's located in that phagosome itself. In terms of maintaining the pH, most papers assume that this prevents phagosome-lysosome fusion. I didn't really see any good evidence for that being the case. Some papers also think that there is some unknown, unelucidated mechanism that the yeast uses inside the phagosome itself in order to actually modulate this pH. But I think I'm pretty comfortable in saying that those mechanisms are not well elucidated at this point. Another virulence factor mentioned before is the changing of the cell wall to high-dose beta-glucans with alpha-glucan, and that, of course, is going to reduce recognition from innate immune cells, specifically macrophages of this organism, which is going to reduce anti-inflammatory responses by those macrophages. So yeah, those are a few virulence factors. Now, once you actually get infected with this organism, what is that going to look like? Well, for most people, it's not going to look like anything, which is good. For most people, you're going to have some flu-like symptoms, chest pain, chills, fever, that kind of thing, and then it's usually self-limiting, which is nice. So that is your general acute histoplasmosis. You, of course, could be completely asymptomatic as well, where you have no symptoms. Or you could also present with pneumonia or pericarditis. There are two other kind of well-characterized forms of this. The second is disseminated histoplasmosis. So this is common in immunocompromised individuals, though it was also reported in 29% of normal non-immunocompromised patients. And this is the most lethal form of this infection. At this point, it's disseminated because it's in your macrophages. They go to your lymph nodes, they go to your bloodstream, and then they disseminate throughout your body. You can get bone marrow toxicity, splenomegaly, hepatomegaly, and skin and mucosal lesions. And so this is a really good figure showing these lesions in AIDS patients. You can see here, I believe this is the lung parenchyma, where you can get this cavitation of the lung. Here are some lesions. These are internal mucosal lesions. You can see those here as well. Yeah, so it can be pretty gnarly. The other clinical presenting symptoms are chronic pulmonary histoplasmosis, and this is usually common in older patients who have had previous lung damage. You can see here more clearly this lung cavitation, and that happens because you get pulmonary infiltrates that turns into scar tissue, i.e. fibrosis, and then that eventually leads to cavitation. Interestingly, this presentation of symptoms usually is confused with tuberculosis, actually. Another thing, if I don't mention it later, I'll mention it now. This is a severely underreported disease in the world. There's around 100,000 cases worldwide, but there are some countries where it's not even reported in. And most of the tests looking at endemic areas were done very surveys were done a long decades ago. So the information we have about this organism is very dilute and I would say outdated at this point. Epidemiology, yeah. So like I've kind of indicated before, anywhere where you're going to have avian/bird droppings that is at risk, basically. It's not really specific human populations. It's not specific ages. It's if you disturb something and it has this microconidia in it, and you inhale that microconidia, you're at risk. So frequent outbreak settings are buildings, chicken coops, farms, contaminated outdoor areas. 77% of outbreaks between 1938 and 2013 had evidence of bat and bird droppings. So that kind of just solidifies that link. Of course, there is a link between environmental disturbance and outbreak. So a lot of construction workers and that sort of thing who are disturbing environments which have been contaminated with fecal matter from infected birds and bats, they are severely at risk. And even in some cases where they use proper PPE as wanted by NOAA or OSHA, I'm sorry, they still reported being infected, and that's probably just because they weren't wearing their PPE properly. Again, there's 100,000 cases worldwide per year, but this is severely underreported and in some countries isn't even reported on. In HIV and AIDS patients slash immunosuppressed individuals, this can take root a lot easier, and it can also be transmitted via organ transplantation, though that's pretty rare. In terms of diagnosing this clinical illness, the gold standard is recovery of the organism from culture, though this, of course, takes quite a while and is not the best way of doing it. If I was in a clinical setting, I would probably use PCR or some sort of antibody detection assay. Other traditional approaches are histopathological staining, and you can see a great example of that here. This is a little neutrophil right here, and these two kind of oval, multicolored ovals are the yeast form of a cell that have actually penetrated into this neutrophil. And so this is pretty commonly used. Another commonly used assay is antigenic detection assays. So it's basically an ELISA. One of the common ones is MiraVista, and that looks at the galactomannins in the cell wall. For some reason, this isn't available commercially, and there are some commercially available assays that have been looked at, though some of them, the papers indicated that they weren't as good as the MiraVista, or they had more cross-reactivity than the MiraVista, because the MiraVista is looking at galactomannins. This is found in quite a few different fungi, so cross-reactivity and false positives are possible. And then, of course, the more modern nested and qPCR techniques looking for specific genes from this pathogen, amplifying those. If you amplify it, you got it. So that one I think is the best one, only because it's so fast. Designing your primers is really easy. You can get those pretty quickly, and you can make them unique to specific genes that would only be found in this organism, which makes it much more selective rather than this Miravista, which can cross-react and give you false results. The treatment is pretty simple. And like I said, in most cases, it is a self-limiting infection. But in cases where it's disseminated or where there is chronic pulmonary infection, that sort of thing, they will use antifungal treatments. The most common is intracanazole, and this treatment can last from 3 months to a full year. So you're usually taking it twice daily for a minimum of 3 months, all the way up to 1 year. So the treatment can last quite a while. Amphotericin B is also used in conjunction with intracanazole. And those are pretty standard treatments for this organism. I wasn't able to find that this was an organism which had developed well-characterized antifungal resistance. So I kind of just want to look at the mechanism of action for itraconazole. So in this case, itraconazole inhibits ergosterol synthesis. Ergosterols are embedded in the membrane of the fungi, and it kind of promotes membrane stability and that sort of thing. So this molecule here, this is the actual itraconazole itself, will bind to this 14-alpha demethylase protein, and then that is what actually prevents ergosterol from being synthesized. In very rare cases, it can lead to cardiotoxicity, and indeed, some people have had to receive transplants because this did not react very well with them. Luckily, that is rare, so we don't really see that in most clinical patients, but it is possible. There was a few papers from the 90s that looked at AIDS patients, and one of the antifungals which it developed resistance against was fluconazole. And this paper essentially just showed that there was lower ability for this strain, this variant of histoplasma to be killed with this, but the actual mechanisms of the resistance weren't really well characterized. It was very much just like indirect evidence of this drug resistance. Either way, like fluconazole was not really used like intracanazole was. So yeah, there wasn't really much in that, which I guess is good, but that also means that, well, it's probably just not well studied enough. And yeah, so that was Histoplasma cannulatum. It's a pretty interesting organism. Yeah, I hope that you guys learned at least a little bit. I would ask if you had any questions, but I won't really be able to hear them or answer them. So I hope that you have a fantastic day, and thank you very much.